Welcome. It's my special pleasure to welcome you for another round of the Bertalanffy Lecture, sponsored by the Klaus-Tschira-Stiftung at the Center for Organismal Studies, but as you can assume, not in our beautiful Bertalanffy Lecture Hall, but rather online on Zoom. You see one reason why we don't have it in the lecture hall. It's still under renovation and likely to be ready maybe around May next year. And we're going to meet there in a year from now or half a year from now. But what we have, and I'm very glad that this has worked out, we have Elaine Ostrander here presenting us, I think, a really fascinating story about dogs. I'm really glad that we could win Elaine Ostrander to give that special presentation. And I'm really happy and proud that this worked out, because I'm absolutely convinced that Elaine is the person to, well, relaunch the series. Why do you think that? Because the way she's doing science is in a way almost citizen science. She works with dogs, and she found a way to collaborate with dog breeders and dog owners that they are really part of the science she's in the end performing. And after her presentation, you will understand what I'm talking about, and maybe you're tempted to contact her and offer samples of your dogs, but that's something you can maybe discuss with her afterwards. Elaine has been studying and getting her degree in Oregon. After that, for her postdoc, she went to Harvard, and after the postdoc in Harvard, she went back to the West Coast, and after many stations, eventually ended up at the National Institute for Health, NIH, in Bethesda near Washington, DC, where she's ever since, and running a really exciting program on, well, mammalian genomics in the widest sense. So by studying genomes, she understands the function of the genome, be it in normal development, but even more importantly, also in disease, and how dogs can tell us many things that, well, you wouldn't easily learn if you're studying humans. And fish, well, they're too small and too distant and not furry enough to be of relevance for humans. So I think without any further delay, I would ask Elaine to start sharing her screen.
Well, good morning, everybody, and thank you so much for coming and giving me this opportunity to share with you some of the work that I and an amazingly talented group of students and postdocs and undergraduates have been doing in the last several years towards understanding all things about dogs. So in this slide, you see a standard poodle, and she's all poofed out to get ready to be groomed for a dog show. And you can see her 5 adorable little puppies there. So I've tried to make this talk accessible to people at all levels of education. Some of the slides are a little busy, but just focus on the big picture. Don't get worried about the details. And all the slides have cute dog pictures on them. So those are certainly there for your entertainment as well. So let's go ahead and let's dive in. Now, the first thing people always ask me is how and when were modern dog breeds created? Well, dogs originated in the Old World and they're sister to the Eurasian gray wolf. Modern dogs, so all those dog breeds you see running around in the dog park, are from an ancient, now extinct lineage. So dogs pre-domestication, or wild canids pre-domestication, and modern dogs today, that link between them is from an ancient and now extinct lineage, not here on the earth today. Dogs are the only large carnivore to be domesticated, and you'll be surprised to hear that that took place 15, 20, at the most 25,000 years ago. So evolutionarily, that is a teeny, 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 tiny drop in the bucket. And that's going to work in our favor as we start learning about the dog genome and trying to figure out where various genes responsible for various traits are located. Now, there are lots of population bottlenecks that are associated with domestication and breed formation. So a population bottleneck occurs when a limited number of individuals are contributing to the gene pool. It might be a popular sire who won at some big fancy dog show. And now everybody wants to breed to that individual. And so their genome through their sperm is now overrepresented in the population. It can happen if there's a big die out and there's just a small number of individuals responsible for creating the breed, much as happened after World War II. So lots of population bottlenecks, and those work in our favor as well, as you'll see in the course of the seminar. All right, these are some of my favorite breeds this week. I hope you see your favorite breed on this or on another slide. So I put this slide up to remind all of us that when you look within a single breed, there's a tremendous amount of homogeneity or sameness. And when dogs from the same breed are bred, they produce progeny that look pretty much identical. The same is true of their genome. So if you look at the genome of all of these Dalmatians or all of these Basset Hounds or all these Bernese Mountain Dogs, you're going to see tremendous amount of similarity, or what we would call homogeneity, sameness, same order of base pairs. And yet when we look between breeds, not only is their appearance very different, but their genome is increasingly different as well. And so we say there's a lot of heterogeneity when we look between breeds. Homogeneity within breeds, heterogeneity across breeds, and that includes both their appearance and behavior, as well as their genomes. Some things to know before we dig in today. All domestic dogs are members of the same species, and that always surprises people because they look so different, they behave so different, but actually they're all members of the same species called Canis lupus familiaris. And for the most part, they can all be bred to produce fertile offspring, and that surprises people too. But in fact, it's pretty much been done with all different dog breeds, you know, all 350 of them across the world, and indeed it's true. Now, my lab doesn't breed any dogs or keep any dogs in kennels or direct any breeding. We don't do that stuff. What we are is in a long-term partnership with dog owners, dog breeders, dog lovers, veterinarians. This truly is a citizen science project. We interact with all of these people. We talk to them about our projects, and normally they are more than willing to provide us with DNA samples from their dogs. And we have in our freezers right now about 36,000 dog DNA samples. And that's really a testament to the success of citizen science, where scientists work directly with people because we have a common goal. We really want to improve the health of dogs, and we want to learn stuff about them. I mean, how can these all be members of the same species and yet all look and behave so differently? So as you saw on this slide, breeds display extremes of morphologic variation, but they also have a lot of variation in how they behave. Think about herding dogs and draft dogs and guard dogs, all those sorts of things. Not surprisingly, because there's been strong selection to get dogs of a particular look, aesthetic, or a particular set of behavioral traits. Along with that selection has come disease susceptibility. So many breeds have within that breed an excess amount of a particular disease or an excess of disease susceptibility. So something like the Bernese Mountain Dog at this bottom left corner, 25% of those dogs will get and die of a kind of cancer called histiocytic sarcoma. And yet you don't find it in the Dalmatians at all. You don't find it in the Basset Hounds at all. So we see these extremes of disease susceptibility because as humans, we have selected so strongly for a particular aesthetic and particular behavior patterns. I could give an entire lecture about disease susceptibility, but I decided not to do that today and instead to focus on morphology and a little bit on behavior because I thought that would be the most fun for us. And finally, the structure of a given breed results in long stretches of what we call linkage disequilibrium. So simply put, that means because individuals are frequently used over and over in a breeding program, uncles and nieces are crossed, grandparents and grandchildren are crossed, and so on, in order to get that perfect prize winning dog. There are stretches of the genome that go for long, long distances, millions and millions of base pairs scattered throughout the genome, where there's similarity or identity across every single dog of the breed. So for Dalmatians, there might be really strong identity across the genes that are important in black spotting, because the selection for black spotting has been so strong. And that also plays to the strengths as we go forward and we try and understand what are the genes responsible for particular traits. So I won't use the term much, LD or linkage disequilibrium, but just know in the back of your mind that, you know, within a breed, there's a whole lot of sameness, and this is another way of measuring that sameness. All right, this is my favorite slide in the talk. So this is work that Heidi Parker has done, and this is her dog, Grace. And everyone in my lab has a dog, by the way, or two, or three. Heidi has three. And so what Heidi has done is she's asked the question, how do dog breeds relate one to another? What got mixed with what to make what? And she's done extensive studies published in really high profile journals like Science and Nature to address that question. And this is one of those. She published in 2017. She's about to publish another one now. So what she did is she took 161 different dog breeds, and she took 10 dogs of each breed, got DNA samples, and the breeds are indicated for you here in this giant circle. And then she tested those dogs at 170,000 random points in the genome, just random places in the genome, and said, what base pair is at that position? And then she took all of that and put it into a bunch of computer programs and said, how would you draw the most parsimonious tree? You know, what best matches what in terms of understanding the structure of modern dog breeds? And she ends up with these 23 color-coded clades, and dogs within a clade are particularly related. So over here in this light aqua, Australian Terrier, Silky Terrier, Yorkshire Terrier, Norwich Terrier, Norfolk Terrier, and so on and so forth. Down here we have hounds, Basset Hound, Red-Bone Coonhound, Foxhound, Otterhound, Bloodhound, and so on. And so we didn't tell the computer program what we thought should be related one to another. We simply gave it all the DNA profiles and said, how would you draw this? And it's really beautiful. It comes out just, it's the kind of data you look at and you kind of go, oh, that is so pretty. And we have posters of this up in our lab. And we're ever expanding it, especially with dogs, more dog breeds from Western Europe, from Southeast Asia, and now from South America as well, and hoping to get some from Africa. And we now have 225 breeds. As you can imagine, this has become much more complex, but we still only have these 23 color groupings. So that's pretty cool. Shows what we've been doing is right. Now, why do we care about this tree so much? Because we use it for every experiment that we do. So let's say a Boston Terrier comes into my office and it has epilepsy. Now, lots of dogs get epilepsy, lots of humans get epilepsy. In humans, it's been really hard to find those genes because people don't come with signs that say, okay, this group is this gene, this group is that gene mutated, and so on. And yet we know there's, 50, 100 genes, however many there are. Dogs do come with a sign because this dog's a Boston Terrier. So I can reasonably hypothesize that if a boxer or a bulldog or a dog de Bordeaux or a bull terrier, min bull terrier comes into my office with epilepsy, they're going to have a mutation in the same gene at the same position because they share recent common ancestors. And that's what this little tree structure here is telling you. All these dogs here in dark blue share recent common ancestor. Now, on another day, a Scottish Terrier comes into my office with epilepsy. Well, I can reasonably hypothesize that an Australian Terrier, a Silky, a Yorkie, a Norwich, a Norfolk that came into my office with epilepsy would have the same mutation in the same gene as that Scottish Terrier because they also share recent common ancestors. You can imagine a little tree here and see how close everything is clustered. it's way on the other side of the wheel is what we saw in these bright blue ones, telling us that this gene and mutation that's a problem in the terriers is probably different than the gene and mutation over here in these bully breeds. And so we can do this and do this and do this, and we can find lots of genes responsible for epilepsy, much easier and much more efficient than trying to figure it out in humans where we just don't have this kind of population structure. And so we're interested in doing that. And this is why human geneticists have suddenly become very, very interested in dogs as well. Okay, now we can do other things with that data set. I told you we had data at 170,000 random places in the genome. So from that, we have computer programs that will build a haplotype. This is one of just a couple of words I need to define for the talk. So a haplotype is simply a group of DNA variants or changes from the canonical sequence on a particular chromosome, and it's inherited together from parent to offspring, down, down, down. So it's going to be a group like the linkage disequilibrium of hundreds or even thousands of DNA variants that segregate together. So if one of the parents has it, you'll find kids who have it on one of their chromosomes, that same pattern, and down and down and down. We can also use that data and we can look at sharing between individuals. And this all works because, remember, previous slide, things are inherited from a common ancestor. And here's the kicker. The longer the haplotype is, the more recent the ancestor was. And if you think back to your studies of mitosis and meiosis, that would make sense to you, because the more meiotic events you go through, the more opportunities there are for rearrangement, right? Whereas the longer the haplotype, there haven't been as many opportunities for rearrangement, telling us that everybody's much more closely related. The ancestors are much more closely related. And we can see that here in this slide. When we look across clades and we ask, how much homozygosity is there? We get this green box. When we look within a clade, so we're looking at different breeds, but they're all related, we see the amount of sameness or haplotype sharing increases. But it's really when we look within a single breed that we see the greatest amount of haplotype sharing. And that makes sense, right? They're really closely related. They've had those population bottlenecks, and that's why there's going to be an excess of sharing. So it all comes back to those lectures you got in science on meiosis and the recombination that occurs during meiosis. All right, why should you care? Well, first of all, if you're a dog aficionado or lover, like half the people in my lab are, I can tell you something about when your breed was created. So we use that exact same data. We like to multi-purpose data in the lab. And we can figure stuff out, like the Norwich and the Norfolk Terrier. They were divided into separate breeds as we look down on the x-axis about 30 years ago. The Eurasia breed began with a Chow Chow cross, and that happened about 50 years ago. Irish Wolfhound had to be recovered using the Scottish Deerhound and the Great Dane, and that happened after World War II. These big dogs just didn't survive the war, so that happened about 90 years ago, and so on and so forth. And we can even figure out really complex scenarios. Bulldogs, French Bulldogs, American Staffordshire Terriers, all came together to create the Boston Terrier about 150 years ago. So DNA is really powerful. It doesn't take much in order for us to learn a lot about how a given dog breed was created. Now, we published a paper in Science in 2007 where we mapped the first big trait in dogs, and that was body size. So what makes big dogs big and makes little dogs little is evidenced by the Great Dane and the Chihuahua here. And since then, we've mapped lots of genes. We found other genes for body size, genes for skull shape, and that pushed in nose that you see in things like bulldog is called brachycephaly. Genes for leg length, genes for coat color, fur texture, for length, for growth patterns, for loss, for the 50% of people in the audience who want to keep track of what those genes are. And each one of those has been a fun, exciting project that one of our trainees did. So one of the first ones we did is chondrodysplasia. So these are these really short legs on these proportionately normal bodies. It's a breed-defining trait for about 20 domestic breeds, and you'll recognize the Basset Hound and the Dachshund. You'll probably recognize the Corgi. You might recognize the Scottish Terrier because I always win at dog shows. You may not recognize the Dandy Dinmont Terrier because only about, I don't know, 20 of them are registered each year, at least in the United States, probably more elsewhere. So Heidi said, what's the gene? What's the gene or genes that are responsible for that? So here's some bones. I love looking at bones to just give you a mental picture of this. So on the right is a normal set of bones for leg bones. And on the left are the bones from some of the dogs you saw or breeds you saw on the previous slide. And for these bones, you can see that they're very heavy, they're very curved, they have big fat joints. So in these dogs, if you look at the Basset Hound, you'll see that the feet are turned out. All right, so these dogs don't have any other health problems, and the mutations believed to have arisen about 500 years ago. So what we did is something called a genome-wide association study, and I'll show you lots of these. They all look pretty much the same. They're organized the same. So on the y-axis is a measure of significance. It's a p-value. And on the x-axis are each of the dog's 38 chromosomes, autosomes, in addition to the X. So chromosome 1, 2, 3, 4, 5, and so on. So we took a bunch of dogs who had these short legs, and then we took a bunch of dogs who had normally proportioned legs, and we took those same 170,000 points of variation, and we said, where in the genome are there statistically significant differences? And it shows up right here on chromosome 15. And when we say significant, we mean significant because we had 10 to the negative 102. So imagine 0.102 zeros and then 1. That's how significant this result is. Probably one of the highest p-values ever reported in the genetics literature. So we knew exactly where the mutation is, and we actually know what it is. So I'll tell you what it is. It's something called a retrogene. So in a retrogene, what happens is, you know how you have exons and they're the coding part of the gene? And in between the exons, you have introns and they don't code for anything. Well, in a retrogene, those introns are gone and the whole gene has already been zipped together and put somewhere else in the genome. And so that's exactly what's going on here. There's the normal copy and then there's the retro copy. And because the retro copy doesn't really know what to do with itself, it doesn't have any promoters or enhancers, it just takes signals from whatever is around it. And so if you look at this indicator here, in a non-chondrodysplastic dog, in this particular assay, you see these two bands here, whereas in chondrodysplastic dog, you see the normal gene, but you see this extra band up here, and this is a sign that we have a retro gene in addition to the normal gene. Now, we don't know exactly how this causes the trait. We think that the overexpression or the mistimed production of the protein from the retro gene leads to early closure of the growth plates in these chondrodysplastic dogs. So the dog can grow normally, except the legs, growth plates are closed, so they can't elongate. So that was pretty cool, and we published that in Science as well. Now that all went along swimmingly, and we identified genes for lots of traits until Joss Plessé joined the lab, and this is his dog. And that's Joss, obviously. And Joss said, I don't want to map things one at a time. I want to map everything at once. Joss likes to think big. So he put together a data set of 538 domestic dogs from 144 breeds. Some of them are shown for you up here. He also had 54 wild canids, and he had 104 village dogs. And then he said, I'm going to look at all that, but I'm going to look at it by direct DNA sequencing. I want to sequence each one. And when he did that, he found 91 million points of variation. And he said, that's what I'm going to use to find genes for everything that there is. So first he looked at furnishings, the mustache, presence or absence of a mustache and eyebrows that you see in the miniature schnauzer, but you don't see in the miniature pinscher. And we got this great signal in a gene called R-spondin2, and it's got hundreds of extra base pairs at the end of the gene that's responsible for this trait. He looked at fur length. Afghan hounds compared, for instance, to whippets. And he found a mutation in FGF5 on chromosome 32. And again, really significant, 10 to the negative 19. But then he looked at harder stuff like standard breed height, standard breed weight. And you can see that there's lots of genes that are responsible for each one of these. And we expect that. It's a more complex trait, it's a harder problem, and there's lots more variation. It isn't a yes-no kind of thing. So he looked at some of these in detail to see if he could figure out what the mutation is. And one of those is a gene called L-CoRL. So L-CoRL stands for ligand-dependent nuclear receptor co-repressor-like gene. It's a regulatory gene. And what he found was that there was a one base pair insertion in the coding region of the gene that was completely absent in little dogs. It was in about 20%, 15, 20% of medium-sized dogs, and we found it in 80% of big dogs, and that is very statistically significant. Now, what's interesting is L-CoRL comes in two flavors, a short flavor and a long flavor. They do different things in the cell. And this mutation was present or absent in the long flavor, the one that's 1,831 amino acids. And what happens? Well, the same gene and these same two flavors are in humans, and obviously in all dog breeds. And what you find in this long version is if you have the mutation, like the big breeds do, you lose a third of the protein. That one base pair change knocks out a third of the protein. And that's what's contributing to these dogs being so big. Don't see it in the medium-sized dogs hardly at all, and we don't ever see it in the tiny breeds. So that was pretty cool. So this is called a heat map because of the color coding. And this is probably the most complicated slide I'll show you, I promise. So what we have down here are, in each column, are the names of a bunch of genes we found that control body size. And on the y-axis, we have standard breed weight. So how much those breeds weigh on, different breeds weigh on average. We didn't specify which breed, but lots of breeds. And I apologize that this is in pounds. My others were all in kilograms. And then we said, do you or do you not have the mutation? And the color coding tells you if this is a mutation you only see in dogs, and then it's going to be dark red, or is it one that you primarily see in something like wolves, an ancestor to dogs? So when we look at small dog breeds, 5 to 10 pounds, 10 to 15 pounds, they have their own unique mutation that's largely in red, except for the couple columns here. And as we get bigger and bigger and bigger dogs, that drops off, and we're seeing more and more and more yellow. We're seeing a lot more sharing with wolves. There's always an exception. These are Rottweilers. I don't know what's going on with them yet. I still have to figure that out. And then it flips when we look at these two genes, ACSL4, IGSF1. What they do is not important for a minute, but they're on the X chromosome. And then it flips because now we're seeing a new mutation in big dogs that's absent in small dogs for IGSF1. And we're seeing a new mutation and occasionally for ACSL4 in big dogs, but never in little dogs. So what's going on and why do people care so much? Well, in IGSF1, the dog or the derived allele is in 90% of large breeds and a subset of medium breeds, and really none of the small breeds. We know this gene is associated with being heavily muscled. In humans, we've figured out already that's involved in the thyroid hormone pathway, and when mutated in humans, it's associated with obesity. This makes sense. Breeders are selecting for these big, big dogs And the same gene, when mutated in humans, is responsible for a disease. So now human geneticists are really paying attention, because just by looking at these traits that breeders are trying to put in dogs, we're learning something about human health. The same is true of the other gene. It's responsible for a bulky phenotype characterized by back fat thickness. We see it in only a subset of the big breeds, things like the English Mastiff, which is very bulky, but not in the Great Dane, which is very lean. It plays a role in fatty acid degradation, lipid biosynthesis, so fat metabolism. In humans, when mutated, it's responsible for resistance to insulin, leading to obesity. Again, another example of the same thing. So we found the genes that account for more than 90% of size variation in dog breeds that we tested. And what's interesting is that number of genes is, maybe 20-ish. In humans, if we were looking for the genes responsible for being 5'5" versus 6'5" and everything in between, there are hundreds of genes, hundreds. And yet the difference between being a Pekingese and a Great Dane, we're only looking at 20 genes or so. So the reoccurring theme here is that there's small numbers of genes or loci of large effect in dogs. And that goes back to those bottlenecks and that really, really short evolutionary time. Whereas in humans, it's the exact opposite, much bigger evolutionary time, lots more meiotic events. And so we see large numbers of genes of small effect in humans. Which one's easier to study? Oh, the dogs. Rather look at 20 than have to look at hundreds. So this gives a scaffold or framework for them thinking about dogs, or thinking about humans. Same is true of ears. Somebody in my lab got interested in drop ears, like you see in the miniature pinscher versus the beagle. As you see in the miniature pinscher and beagle versus the Eurasier and the French bulldog. They found a really strong hit on chromosome 10, and the gene is MSRB3. In humans, when mutated, it's associated with deafness. So it makes this aesthetic trait in dogs, in humans associated with deafness. Large versus small ears, these two big ears, these two small ears. And there are a couple genes it could be, but the one it is called a KCNQ5, also involved in deafness in humans. It plays a vestibular role in the inner ear. Same story, an aesthetic trait in humans, in dogs, and a disease in humans. I don't know why there's a yellow line there now. And if you have infinite money, you can go and get your puppy tested, and they'll look for the presence or absence of the mutation, and they will tell you your puppy's going to have normal prick ears, large prick ears, normal drop ears, or large drop ears. And you can do that if you have infinite amounts of money to spend on your puppy. Okay, so the next thing that we looked at is something I like to call the champion's genome. And this was done by Jae-Min Kim. And he just wanted to know, what are the underlying genetics that makes sport hunting breeds, pointers, retrievers, very active and athletic dogs, shown on the top panel, different than, for instance, terriers, small, energetic, aggressive, originally bred to hunt vermin. And so he sequenced every base pair of 21 sport hunting breeds and 28 terrier breeds. And then he said, what are the differences? What's going on here? And I would have bet the farm that what we were going to find were all genes important in the nervous system. They do different things that's got to be controlled by the brain. I was wrong. The most important things were things that were associated with blood flow, endurance, cardiac development. Physiology trumped everything. So here are some of the genes. Here's what they do. Vascular muscle cell contraction, muscle-specific actin binding, resting heart rate, skeletal muscle contraction. Now we broke it down a little bit. We went to the track, we didn't tell NIH, and we got DNA samples from racing whippets. And there, the most important gene was TRPM3, and that's responsible for muscle cell contraction. And that makes sense given what these dogs do. We had found mutations also responsible for musculature in a gene called MSTN1 or myostatin1. Together, these account for 24% of racing speed in whippets. Two genes, 24% of racing speed. That's incredible. In humans, much more complex. And this is what the myostatin mutations look like. In the first column are normal whippets, and in the second column are heterozygotes. They have one copy, and you can see they're much more muscly. And in the third column are dogs that carry 2 copies of the mutated gene, and they're just grossly over-muscled. So if you look like this, you're going to have a big advantage at the racetrack. All right, and we also looked at agility. So that's like ninja warrior kind of stuff. And again, there was a single gene RYR3 important in skeletal muscle contraction. That makes sense. You run forward, you run backwards, you turn around, et cetera, et cetera. So this was actually a really fun study. Some of the genes were particularly interesting, like ASIC3. That's important in resistance of skeletal muscle to fatigue. So this is a neural gene. It's located on neurons, and it's critical to the perception of pain. So when you look in mice, what it does is it prevents the perception of pain. So physiologically you hurt, but your brain doesn't recognize it. And so you don't know that you hurt. And it's actually fixed in all sport hunting breeds. They all have it, whereas none of the terriers have it. And that result is really statistically significant. Another interesting one is CDH23. When mutated in humans, it's associated with a syndrome called Usher syndrome 1D. Now, Usher syndrome is a developmental disorder, and kids who have it have lots of traits, but not infrequently they have hearing loss. That's kind of surprising. Why would our dogs have a mutation that might cause hearing loss? Well, they really did. This mutant allele was really common in sport hunting dogs and virtually non-existent in terriers. And if you look at predictions for how it affects the protein, that mutation is pretty deleterious. And if you look over here on the right, you're looking at DNA sequence from a bunch of mammals: pigs, cattle, sheep, pandas, ferrets, dogs, etc. And at this position colored in yellow, they all have a valine, except for our sport hunting dogs who have this mutation. And then everybody else has a valine. So I didn't really understand this. And so I went out and I talked to a bunch of hunters and said, can you think of any reason this would be true? And they said it most definitely is true. Deafness is pretty common in these sport hunting breeds. It virtually never occurs in terriers. And their hypothesis is that it reduces the startle reflex. So if you're not totally deaf, you don't really perceive all the background stuff. You're not distracted by, the birds cawing and quacking and whatever else it is that birds do, nor are you freaked out when the gun shoots off. And so these dogs who have their hearing ratcheted down actually do better than dogs who are super hyper hearing and hyper perceptive. So I got to work with some deafness people and figure out if that's true, but I thought it was a really good hypothesis. All right, so this is my last vignette, and we're going to change gears here. And I'm going to tell you about New Guinea singing dogs. So New Guinea singing dogs are arguably the rarest breed in the world. There's only about 200 to 300 that are alive today, and they live in zoos, they live in conservation centers, private rescues. Here's a picture of one here. They're average looking, 10 to 20 inches tall, 20 to 40 pounds. They have a short, dense coat. They have prick ears. We know why now. They have a tail that curls over their back. My new postdoc will know why. Now, while their appearance is very commonplace, their voice is not. And this is why they're called singing dogs. So this is a very haunting noise. Imagine being at a campsite and it's all dark at night and you hear this very haunting sound. It's called singing because it has aspects of singing. It goes up and down a register. It has tonality, all those things that people who study voice know about. And so the thought that these would be lost from the face of the earth was really, really devastating to us. This particular New Guinea singing dog is part of a population of that 200 to 300 that's in the San Diego Zoo. So where do they come from? Where do those captive dogs at the zoos come from? Well, in 1956, there were a pair of so-called mountain dingoes. They were captured and they were sent to a park in Sydney. And they got the name New Guinea singing dogs because they were from New Guinea and they had that singing quality to their voice. The female was pregnant and she had 4 puppies, 1 male and 3 males. And they're actually the primary source of all the New Guinea singing dogs that are in the US today. Now, 20 years later, another 5 live specimens were captured and they were sent to Germany. And that was the last capture and exportation of New Guinea singing dogs. They hadn't been seen in the wild since 1976. So those 8 dogs are the ancestors of those 200 to 300 captive dogs. That's it, those 8 dogs. So you can imagine they're really inbred. That's a population bottleneck. Not observed otherwise in the wild since the 1970s. And these are pictures from dogs in the various conservation centers. So we set up a collaboration with just a brilliant field biologist named James McIntyre. And in 2018, he led an expedition to Papua, Indonesia. And he saw what he believed using these night cameras were New Guinea singing dogs. Now, he called them Highland wild dogs at that time because we weren't sure at the genetic level, but he had good sight footage and he had good sound footage as well. They were found near a gold mine in the foothills of Papua New Indonesia. And so he was able to catch 2 males in a bait trap. He sampled them, he tagged them, of course they were released. And then there was 1 female that was recently deceased that he found, and he was able to sample her as well. So he sent the samples to us, we extracted DNA, we tested those samples at 150,000 random places in the genome using the same technology Heidi had used years ago. We merged that data with data that Heidi had generated on that first data slide you saw. It included 1,350 breed dogs, 161 breeds, just like you saw before. But this time we also threw on dogs, village dogs, dogs that hang out outside by the garbage dumps from China, Vietnam, village dogs from New Guinea, Polynesia, Fiji, Borneo. And then we threw on some from Namibia kind of as a control. And then we put in some of the captive New Guinea singing dogs, and then these 3 so-called Highland wild dogs that we wanted to see if they were New Guinea singing dogs as well. And again, these are our dogs from the conservation center. They're ones we saw in the wild didn't look nearly this good. All right, and so I know this looks busy, but it's so cool. I got to take you beyond it. So we did a bunch of different kinds of analysis. Now, remember, these dogs were originally called like dingo mountain dogs or something because they do look a lot like dingoes. The second panel has a dingo. The third has a captive New Guinea singing dog, as does the fourth has one of these Highland wild dogs, and the 5th is an Akita. And so same deal, we asked the computer program, how would you assign these? How would you divide them? Who's most closely related to who? So at the top here, related to virtually nothing, are all the Western European breeds, like the Labrador Retriever and Golden Retrievers and Spaniels and Terriers, and all those breeds that came out of the explosion of breeds during Victorian times a couple hundred years ago. The captive New Guinea singing dogs are this big red triangle, and they end up being pretty close to the dingo. But look who else is really close to them in this tiny little triangle, because we only had 3 dogs in green, are these Highland wild dogs. So wow, they are closest to the captive New Guinea singing dog. Now, that was awesome. Remember, we didn't expect them to be the same because these captive New Guinea singing dogs have been inbred for decades and decades and decades. And these Highland wild dogs we had sampled from the wild. And then there's a variety of Asian breeds and North America, Arctic breeds, and so on and so forth. So the first thing that we wanted to do was we wanted to know if there really was a difference between our Highland wild dogs and the New Guinea singing dogs and dingoes, because we had a concern that what we were really looking at was some form of dingoes. So in red, we have our captive New Guinea singing dogs. The 3 in green are the Highland wild dogs. So you can see how this is going to end up already. And then everything else are different populations of dingoes: alpine dingoes, desert dingoes, captive dingoes, desert dingoes from 3 different places, blah, 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 blah. And you can see the dingoes divide pretty nicely. And this green over here, this Kelly green, that's a dingo population. It's this dark green here that are Highland wild dogs. And you can see their closest relative are those captive New Guinea singing dogs, establishing that these Highland wild dogs are the captive New Guinea singing dogs. They're more outbred because they've been out in the wild, and the New Guinea singing dogs in zoos and such are more inbred, but we certainly expected that. So we were able to show that New Guinea singing dogs are not extinct in the wild. There aren't many of them. They're very, very rare, but they certainly do exist. And that was so incredibly exciting, and it got lots of media, and it was a pretty cool thing. So we learned that conservation populations of New Guinea singing dogs are inbred. We know they descend from only 8 dogs, and that these recently discovered Highland wild dogs are the original New Guinea singing dogs. How different are their genomes? About 28%. These Highland wild dog version of New Guinea singing dogs have about 28% of genomic material that's absent from the captive population. So that's what's been lost with inbreeding and inbreeding and inbreeding and inbreeding. So that 28% probably includes ancient as well as some modern diversity. And that's the diversity that's lost in the conservation population. So clearly, introduction of these Highland wild dogs into captive New Guinea singing dogs may represent a really unique opportunity to save this rare and exotic species. Lots of caveats to doing that, lots and lots and lots. And I'm not involved in those decisions, and they are complex decisions for a variety of reasons, but certainly under discussion. All right, so I'm going to stop here. I hope I've convinced you that dogs are a great system for studying simple and complex traits, that morphologic studies illuminate genes and pathways that are of interest for human health, and that most traits or phenotypes are controlled by small numbers of genes of large effect, the opposite of humans. We do have a project to sequence, get every single base pair for 10,000 dogs, and making the data all public, and we're at about 3,500 right now. And the genomic analysis of rare populations using the same tools we developed years ago aids in our conservation efforts. So I'm going to stop there. Here are the people who do all the work, and we're very grateful to them, and the people who fund us, as well as the dog owners, especially the dog owners, and of course the dogs, and some of the folks in the lab with their dogs. And if you have a really way cool dog, you can just go to our website, or you can email us at dog_genome@mail.nih.gov. And I'll leave that up for a few minutes. And that's it.
So thanks a lot for a wonderful presentation. As you said, it's clearly citizen science, and I guess there will be, that's a role model. We need more of that. And it can't be all dogs, so maybe eventually things will turn to racing horses or dromedaries or camels. I don't know whatever is bred and where you have these kind of informations. I have questions that are maybe unrelated to your topic, but more related to you and how you ended up doing what you're doing.
Oh, yeah.
We have a large group of, well, young students who are about to develop and see what they do. So how did you come up with the dog idea? Was that something you always wanted because you had dogs when you were a kid? And no. Nothing
So
obvious.
another way to put this is, when I was a graduate student and my first postdoc at Harvard, all my work had been on DNA structure. So topoisomerases, DNA helix, dimensions of the minor groove. I'd had full articles in Science. I mean, that was what I did. And then I switched. I decided I wanted to do biology, and I went to a worm meeting and a fly meeting, because the direction DNA structure was going was crystallography, and I was never going to be a crystallographer. That just isn't how my brain worked. But I never actually had a biology class, still haven't. So I went to a fly meeting, and a worm meeting, a plant meeting, and I got a NIH plant fellowship to study plants, went to Berkeley to do that. And it turned out the lab I was going to was in a state of change. We'll just go with change. And it wasn't going to work out for a new postdoc to join at that particular time. So I sort of advertised myself as a rent-a-postdoc, is what I called myself while I was trying to figure out something to do. Because I had really good skills. You know, I was a whiz at the bench. I could do anything. I could clone anything. I could purify any protein. And so I was in this conversation with Jasper Ryan at Berkeley. And right at that moment, some of these fortuitous moments, papers had come out showing that there were things called microsatellites, and they're DNA markers. They're points of variation. They litter the whole genome. There's one every few thousand base pairs, and you could use those to do genetic mapping. They were polymorphic in populations, but stable enough to follow in families. So they were that perfect thing that evolution had done. And we were saying, you know, wouldn't it be cool? You could make a map of any mammal in the world that you want to study, anything. And of course, the idea of dogs and behavior was front and center. And so I started out there for a year, and then stayed on. And then I went to the national labs where I did that and worked on the Human Genome Project on chromosome 21. And then I went and applied, you know, for various faculty positions, got, I think, all but one of the ones that I interviewed for, and everything took off from there. But I remember very early when I was just starting out as a faculty member, I met Lee Hartwell, who had just won, or was clearly going to, but he Nobel Prize winner for his work on cell cycle regulation. And he said, I want to hear about this dog project. And so I'm telling him about it. It was brand new. I mean, no, you know, and he goes, but you're actually smart. You have all these papers in science and stuff. You have this, why would you do this to a perfectly good career? And I felt about that big. And I slunk out of his office, and then I thought about it. And I was at the Hutchinson Cancer Center, and I had lots of opportunities to work on breast and prostate cancer in humans, which I did with half of my lab. But I remember saying to myself, you know what? I don't really care what Lee Hartwell thinks. I'm going to succeed or fail on my own terms. And if I fail, I fail. And I'll do something else. I'll work on human cancers, and that'll be my career, and I'll get grants, and life will move on. It'll be fine. But I'm not gonna not try, right? And I guess that would be my most important message to you guys is, you know, the only way to succeed is to try, right? And maybe your chances are 5% or 10%, but somebody has to be in that 5 or 10%. Somebody does, so why not you?
Why not you?
And so give it a whirl. See anything that you're interested in and curious in, and do it. Don't do something because everyone else is doing it, right? Don't work on the flying nervous system because you know 25 other people that are doing it, right? Don't just CRISPR stuff because everybody CRISPR stuff in mice, right? Find a biological problem that keeps you awake at night, that you like wander to on the internet when you're bored with whatever you're supposed to be doing on the internet, right? And think about, wow, that would be really cool. Is it something in evolution? Is it something about extinct animals? Is it technology? Is it bioinformatics? How do you take all the sequence and do blah, 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 blah for those of you that are bioinformatically minded, right? And you'll fail a lot of the time, but over your career span, you will succeed. You will make important contributions to science. You will realize that you have to hire people who are smarter than you and who know stuff you don't know. Every person I have hired is good at stuff I do not know. And I would say half of them are smarter than me for sure, right? But that's how you build a team, right? That's how you build a team of experts, and you go forward and you solve really hard problems and you have a high impact in science. If you don't try, none of those things will ever be yours, ever be yours. So that's what I say. Pick something that you care about. Pick global warming, pick anything that you care about, but don't not try. Then you really are failing.
Okay, I think that was the perfect closing remark. So thanks a lot for you staying with
Sure.
us all the time.
Thank you, everyone, for coming and for all the good questions.
Elaine,
Thank you,
that was
everybody.
fantastic, and I'm very much looking forward to seeing you soon again. So that's it.
Thank you, everybody.
And I'll see you latest
Bye-bye.
in half a year when we have the next lecture.
Absolutely. Bye-bye.